Oh, you think carry it around?
Oh, that was Jeremy.
Yeah, I thought camera.
Yeah.
Well, just as good. Always.
Balance. Yeah, you're good. Good. Yeah. Okay, everybody, can we get going? Hi, everyone. If we can just get started. So, welcome to the third Health Science, Health Research Institute seminar series. Thank you all of you for attending and for supporting these events. As I mentioned, for those of you who haven't been to this series before, this started just this year in September, and the goal was to try to cross the all disciplines of health research. So our first one, we had a panel discussion on Indigenous health. The second one, we had Rob Olson, who was talking about his clinical research at the BC Cancer Agency, and today we have Dr. Chow, who's going to be talking to us about his biomedical work on novel medicinal compounds for cancer therapy or others, right, Chow? With our collaboration. So Dr. Lee is a professor in biochemistry and molecular biology at UBC. He's been here for 19 years, I learned today. He has a research lab over in the Northern Health Sciences Center with about 8 to 10 members. And today he's going to be talking about exploring British Columbia's wild mushrooms for novel medicinal compounds. And this is a project he's had going for about 4 years in the lab, and I'm really excited to hear about where they're at with this because, as I said, we're hoping to do some collaboration with Chow looking at these compounds on pancreatic beta cell function. So I think this is a really great example of a project that, while Chow may be interested from an anti-cancer perspective, that it could spark a lot of other interdisciplinary collaborations with other health researchers at UBC. So we're really excited to hear your work. Thank you.
Okay, so first of all, it's great to thanks to Sarah and HRI for inviting me to give this talk. And also great to see that that people from NRES and also health science. So it's great to see that this research draw a lot of interest across disciplines. So that's the whole idea. So today I'm going to talk about how we collaborated with a few faculty on campus to explore British Columbia mushrooms for potential new medicinal compounds. So what I want to say first is, besides doing the research, we got to see the beauty of British Columbia. Okay, that's the best part, I think. Okay. Oops. Doesn't work. Stop moving forward. Of course it does. Yeah. Moving forward.
I don't know.
Okay, so this project that I'm going to talk about, exploring British Columbia wild mushroom, this is a relatively new area to us, so we only embarked onto this about 4 years ago. My domain is actually on nucleic acids, specifically looking at RNA decay, and we have 3 projects related to that. Now, this project, I wouldn't have been able to kick off the research without contribution from several faculty on campus, and which is something which I want to talk about right up front. Okay, so we are an interdisciplinary team. So Dr. Oon Massicott and they are the mycologists, so they contributed to help us find these mushrooms, identify. And we have Dr. Kerry Reimer, who is an organic chemist, who helped us in the chemical extraction of the mushrooms. And Jeff, I don't see Jeff here. So Jeff provided his mouse microcirculation model. So he allowed us to look at the effects of these mushrooms in an animal model. Something I will talk about shortly. Linda is here today. So Linda helped us in collection, vouchering, and also generating a library of these crude extracts. And Maggie is also here. She's involved in various aspects of the project, including supervising students in purifying and identifying these bioactive compounds. Now, students, okay, so they are the backbone of our research. So I want to mention some of the students here. Aaron, he's not here. So he basically pioneered this project. He helped us establish the methods to chemically extract these mushrooms. Ankush worked on the a species called Axylus involutus. So I'm going to talk about that later. And then Samreen Javed, whom Jeff and I co-supervised, worked on a anti-inflammatory compound, which I'll also talk about shortly. And then we have a list of outstanding undergraduate students, especially Gorbin, Jay, and Supreet, who actually conducted the bulk of the extraction and screening of the mushroom extracts for 3 bioactivities, which I will talk about. Farhan attempted to purify a growth inhibitory compound, unfortunately without any success. Taylor, Harry supervised, looked at chemical extraction and screening of some compound mushrooms. Michael did some characterization, and then currently we have 4 graduate students working on purifying, identifying, characterizing some bioactive compounds. And Ahmed, who's currently looking at genetic analysis of our latest batch of mushrooms, and Brandon. Brandon was involved in chemical extraction and screening of compounds, and now he's looking at specificity of some of these extracts against cancer cells. Okay, so without funding, we won't be able to do the research. So I want to say that we're very grateful to the Office of Grants, Research and Graduate Programs to help us kick off this project in the form of seed grant and research project awards to students way back in 2012, 2013. And then we finally got some money from Genome BC and UNBC to allow us to do more research. Then we managed to get funding, infrastructure funding from CFI and BC-KDF. And more recently, we got interest from a industry, Nikkei Canada, who was interested to look at the Japanese weight ratio as compared to BC mushrooms. Okay, so why mushroom? Why not? Okay, so maybe I have to take you back to 2012 when I was doing my sabbatical leave in the Hong Kong University of Science and Technology. So I attended the grad seminar and to understand the type of research that was going on. So it became clear to me that research into natural products are very common in Asia. And also clear to me that a lot of them relied on mushrooms that they buy from supermarket or their friends. So coming from northern BC, we're surrounded by forests. Immediately I thought it would be a great advantage if you can do some research to BC wild mushrooms, because there must be good stuff in the wild mushrooms rather than cultivate. And another reason why I ventured into this is because working on natural product research, there's a likelihood that we can find something that have a direct impact on prevention and diseases. So I did some literature search on medicinal mushrooms. So this is what I found. So it's common area of research in Asia and Eastern Europe, not so much in North America, mainly because pharmaceutical companies aren't that interested in natural product research for a while. So things have changed now. So what I also found was a lot of compounds, small molecules, were from mushrooms and fungi. And this is a short list here. For example, cyclosporin, which is an immunosuppressant from fungi, and then some antibiotics and also anti-malarial drug. This compound called psilocybin, it's from magic mushroom. It's been explored as antidepressant. I don't believe it's been used yet. So these are small molecules. And pharmaceutical company in the West, they're principally interested in small molecules, not large molecules, because those are less well defined structurally and also the mechanism of action. This is a large molecule called polysaccharide crestin. Its structure has been studied but not well defined. So it's from a mushroom called Trametes versicolor, shown here, and has been used. It has been used in Japan since 1970s as treatment for chemotherapy. And it worked very well for some types of cancer, GIT tract, cancers of the GIT tract. For example, gastric, esophageal, colon cancer. But it is not approved in North America. However, it is very refreshing to see that actually there are studies on PSK that's done in the US. For example, this paper shows that PSK can actually stimulate immune cells like T cells and natural killer cells to attack tumor cells. Okay, and it works through the membrane receptor called TLR2. And then there are clinical studies shown here in this paper on the mushrooms on breast cancer patients. Okay, so the studies concluded that the extracts from this mushroom is safe, and it does stimulate to some degree immune system in the patient. So it's good to see that there are studies on mushrooms on clinical trials in North America. Here's another compound, also a polysaccharide, it's a large molecule from mushrooms. And it is also used in Japan as adjuvant for chemotherapy. Okay. Not in North America. Okay, so another reason why we're interested in studying mushrooms because of its diversity. This is a quote from an expert. There are about 150,000 species of fungi worldwide, and only 10% have been identified. So lots to be explored. And another reason why we're interested in mushroom is because there are very few people working on medicinal mushrooms. In Canada, there are only 2 people. They're in Ontario. So they work on medicinal mushrooms obtained from China, not from Canada. And then there are Roger Linnington in SFU and Raymond Anderson at UBC. They're interested in natural product research, but not on mushrooms. They focus on marine organisms. Okay, so in Canada, there are many mycologists, including our 2 colleagues here on campus, but they work on ecology aspects. So there are not many people who work on medicinal mushrooms, especially wild mushrooms. So to kick off our research, here's our hypothesis. So we hypothesized that many unidentified biologically active novel compounds that exist in wild mushrooms growing in British Columbia. So we set out to do the research. So the next 30 slides or so, I'm going to talk about the research that we have done, what we have accomplished. Okay, so the first phase of our research was funded by UNBC, Seed Grant and Research Project Awards. And we collected about 15 species. We genetically identified them, performed chemical extraction, and then we screened them for 3 bioactivities that are relevant to cancer. To identify the mushrooms, there are 2 methods. One by looking at the mushroom itself and then compare it to what's in the book, or use a matchmaker program. But in fact, we really rely on Ook and Keith to make the call, and Linda to make the call. To confirm those species, we did genetic analysis. We isolated genomic DNA from mushrooms, and then we do polymerase PCR to amplify the specific region of the DNA. In this case, ITS2 region of ribosomal RNA gene, and then we subject it to DNA sequencing. Then once we got back the sequence, we blasted against a database online and to identify the species. Some species are easy to identify, but not others. Okay, I won't go into the detail. Okay, here is how we crush the mushroom before we do chemical extraction. So if they are woody, we get the help of John Orlowski. John is not here. John would cut it into small pieces, and then we throw it into a hammer mill, and then we'll pulverize it. And if the mushrooms are soft, we just use a regular blender. Then we subjected them to extraction, shown here. Initially, we did a manual extraction. Subsequently, we used speed extractor, shown here. Initially, we used a Bushy speed extractor, owned by Alan Pettigrew. And later got an speed extractor from our CFI infrastructure. Okay, here's the scheme that we did the chemical extraction. So we do a sequential extraction first using 80% ethanol, and then the filtrate and pass through to 50% methanol, water, and then sodium hydroxide. Okay, at each step, the liquid, we call them, say, extract 1 from ethanol, extract 2 from methanol, extract 3 from water, extract 4 from sodium hydroxide. So later on, I'll basically be mentioning E1, E2, E3, and E4. Okay, so, and then these liquid then evaporated, concentrated, and then turned to powder, and then we resuspend in water and filter sterilize before we test it for activity in cells, in Petri dishes. Okay, so these are the 3 bioactivity that we used, and they're relevant to cancer. We assessed the immunostimulation by measuring a production of a cytokine called TNF-alpha from a macrophage cells. And then we measure the level in the supernatant using ELISA, a method called ELISA. We assess anti-inflammation by simply looking at inhibition of a compound called LPS. We look at inhibition of LPS-induced TNF-alpha production in macrophage. And then lastly, we look at growth inhibition by inhibition of growth on a human cervical cancer cell line called HeLa cells. So this is a result from a mushroom called chaga, in short. The Latin name is Inonotus obligus. Okay, so this is very common. A lot of Eastern European have this, take this. Okay, they know that it has medicinal value, and boy, I can tell you that they do have medicinal value. Okay, it has very potent anti-inflammatory activity. So look at the at 1 mg per ml, even at low concentration, it is suppressing TNF alpha production. So it doesn't have immunostimulatory activity, and it has also has growth inhibition activity. So you can find chaga in the woods, even in the forest for the world. In fact, we picked this one up in the forest for the world. And this table summarizes the first phase of extraction and screening, looking at the 3 bioactivity. Bacillus involutus, Echinodontium tinctorium. I'm going to talk about that later on in a bit more detail. Viola piota aurea. This is something that one of my students is working in the lab, so I won't talk too much about that at the moment. Already mentioned about chaga. Now, Hericium coralloides. This is it's an interesting species. The footnote here says that all the extracts were growth stimulatory. This is one of the only one I know of that has, rather than growth inhibition, has growth stimulatory activity. And this is shown here. So it has actually stimulate growth of cells. It will be of interest, I would say this species will be of interest to researcher who is interested to find compounds that will stimulate cells. For example, people who work on neurological disease or even pancreatic cells. And in fact, somebody in Saskatchewan who's interested in medicinal mushroom contacted me last year and asked me, we have actually worked on Hericium, can't remember the name, Eretnaceous or something like that. It is a species in Hericium, and it is known to be used by patients with ALS. Okay, so I just finished describing the first phase of our research, where we screened about 15 species, screening them for activity. Now I want to talk about the second phase of our research, where we got our funding from Genome BC and UNBC. Okay, so it allowed us to actually collect more samples. We were able to collect 115 samples. Unfortunately, 8 samples were contaminated, so we left with 107 samples, and then we subjected that to genetic identification. Surprisingly, some samples, although they look morphologically different, genetically they are similar. Okay, for example, the 13 samples were Vomitoxys pinicola, 7 samples were Ganoderma aplanatum, etc., etc. So rather than do a lot of repeats, we decided that we'll actually subject 84 species for chemical extraction, and then screen them for the 3 bioactivities that I just mentioned earlier. So these are the places that we went. So we collected the mushroom from Prince George, Smithers, New Hazeltine, Terrace, Rupert, and Haida Gwaii. So in Haida Gwaii, we picked some mushrooms from several locations. I like to do a comparison between Haida Gwaii and Hong Kong, because Hong Kong is place where I go every year or so. So the size of Haida Gwaii is about five times bigger, but the population is very different. 5,000 versus 7 million. Give you a perspective of the scale of the population density. Okay, so we subjected 84 species to chemical extraction, screening for the bioactivities. So 84 times 4, so we got 336 crude extracts. And we screened them for the 3 bioactivities. The next 3 slides, I'll show you detailed data of 3 species, and then after that, I'll show you 3 tables summarizing all the data. Okay, Ganoderma sugae. So this is the most interesting species in terms of ability to stimulate immune system. So this is LPS, which is our control, which is known to be a hammer in terms of stimulating immune system. This species is stronger than hammer. Okay. So it's very exciting to see something like that. And from extract 3, water extracts. And it doesn't have any growth inhibition activity. And then extract 1 has anti-inflammatory activity. So Ganoderma sugae is a medicinal mushrooms. If I have an opportunity, we would like to cultivate this species, specifically from Haida Gwaii, because we found that there are differences in different locations, same species collected from different locations. Lobster mushroom. Rook is not here. Rook loves lobster mushroom. So do I. They smell like mushroom. They taste like mushroom. Yeah, so it's fabulous. So there are plenty of them on campus, by the way, if you're interested. But we won't tell you where it is. It has very potent immunostimulatory activity. It also has anti-inflammatory activity, and anything in terms of growth inhibition, it is very weak. Anything at about 50%, I wouldn't consider potent. So again, lobster mushroom is an edible mushroom. If I have a chance, I would cultivate this. Then finally, Formitopsis officinalis. It is a fungus that is actually highly regarded by First Nation. They actually carved these fungus and put it as a figure in the grave, the shaman grave. So it doesn't have immunostimulatory activity, as anti-inflammatory activity, and also potent growth inhibition. So one of Carrie's students actually is currently trying to see if he can find growth inhibitory compounds from Formitopsis officinalis. So this is this table just gives you a short list of mushroom species that exhibited highly potent growth inhibition activity. Okay, so there are 21 species, but obviously I'm just selecting a few here. So we found that there are 25% of the species exhibited potent or moderately potent activity. So 25%. So the bottom line is there's no shortage of bioactivities that are exerted by these mushrooms. There are many of them, they do. It's a matter of pick and choose which one you want to work on. Okay? So we pick them based on novelty, abundance, availability. So For example, Albatralis flatty is something that one of my students is working on, Thomas, over there. And Phomitoxis officinalis, Terry's student is working on it right now. In terms of immunostimulatory activity, there are about 23% of the species show potent activity. The one colored in red are the ones that my students are focusing on right now. Finally, this table shows you the selected list of highly potent anti-inflammatory mushrooms. 40%. Okay, actually, there are more mushrooms that have anti-inflammatory activity. And the one colored in red, especially this, E. tinctorium. I'm going to describe a bit more detail of anti-inflammatory polysaccharides, that one summary we worked on. Okay, so the next few slides, I'm going to focus on a bit more detailed studies by my students who, so they basically look for these compounds. They do purification and attempt to identify these compounds and then characterize these bioactive compounds. So I'm going to talk about growth inhibitory polysaccharides from Paxillus or involutus and anti-inflammatory polysaccharides from E. tinctorium. So Paxillus, okay. Ankush worked on this species for his MSc thesis. So he found that extract number 4, sodium hydroxide extract, has growth inhibitory activity. This is a growth inhibition assay, dose dependent and time dependent. So extract 4. He then did a lot of experiments to come up with a strategy to purify the bioactive compound. So he used a size exclusion chromatography followed by ion exchange chromatography. This result here shows the dots, the closed dots, shows the inhibitory activity in the expressions. And then the broken lines shows the carbohydrate content. Okay, so this result shows that the bioactive growth inhibitory compound is rich in carbohydrate. These results here just to show that whatever he collected from the chromatography, they're still active. And then I guess Ankush left, and then we have to continue the work, Sebastian and myself. So Sebastian ran a SuperDAX, a high resolution chromatography to show that, so the dots, the black dots shows the inhibitory activity. So, and then we estimated that it's about 229 kilodalton. And then this result shows that the inhibitory compound is rich in carbohydrate, but not protein. So it doesn't correlate with protein. Now, Sebastian also did a biochemical analysis to look at whether the purified polysaccharide can induce apoptosis. So this is a Western blot, just to show you that when you treat the cells with the purified polysaccharides, it can induce caspase 3, a pro-apoptotic protein, and then inhibit the anti-apoptotic protein, an indication that the compound can stimulate cell death in the cells. And then this is just a flow cytometry analysis showing that when you treat the cells with 0.5 mg per ml of the polysaccharides, most of the cells undergo cell death. And then Maggie did the GCG. So this is the data of GC, gas chromatography analysis for those of you who don't know what gas chromatography is, an instrument that can allow us to look at the composition of the polysaccharides, what it's made of, what monosaccharides it is, and the type of linkages. So, so the result shows that the polysaccharide is heteroglycans. So it's made up of many types of sugars, different types of sugars. Glucose, galactose, mannose, fucose, xylose, but no ribose compared to polysaccharide K. And then GCMS methylation analysis shows that the polysaccharide has terminal fucose and glucose. It has branch points at 136 mannose, 126, and then backbone linkages shown here. So I won't go into the details. Okay, so those are the studies on the polysaccharides from Paxillus involutus. Now, I want to talk briefly about Echinodontium tinctorium. So it is it's not an edible mushroom, but interesting in terms of bioactivity. Aaron first worked on this project, found that the extract 2 has potent growth inhibitory activity. Farhan, one of our undergrad students, attempted to purify the growth inhibitory compound without success, but we'll revisit that again now that we're getting better in doing this. It's a learning process. Samreen looked at our past graduate student, looked at the immunostimulatory activity and found that the extract 3 has strong immunostimulatory activity. And currently, one of my students, Macri, is working on finding the compound that is responsible for this. And then Samreen also showed that the extract, sodium hydroxide extract, has strong anti-inflammatory activity. And in the next few slides, I'm going to show you results from her work, part of her MSc thesis. Samreen was co-supervised by Jeff and myself. Okay, so let's look at what she has done. So she actually used 2-column chromatography to purify the compound. What the results here is just for results from the 3rd column chromatography, Superdex 200. So with the help of Sebastian, we show that the anti-inflammatory compound is about 5 kilodalton. It correlates with carbohydrate content, suggesting that it is rich in carbohydrate, but not protein. And then the anti-inflammatory polysaccharide also inhibited LPS-induced nitrite production. So, which agrees with the fact that it also inhibited TNF-alpha, LPS-induced TNF-alpha production, because when you have high level of TNF-alpha, that can lead to increased level of nitric oxide. So Samreen also showed that the E. tinctorium can inhibit histamine-induced TNF-alpha production. And this is a critical experiment because later on she went on to study anti-inflammatory activity in the mouse model, in which histamine was used as an inducer of anti-inflammation. All right, so Jeff is not here, unfortunately, but this is Jeff's domain. Okay, so I'm gonna, I hope he can excuse me if I say something wrong here. Okay, so Samreen decided to look at the ability of those anti-inflammatory polysaccharides in extracts, mushroom extracts that show high, strong anti-inflammatory activity in the mouse model. So we So Jeff established this model since he came to UMBC. So we're looking at 2A arteriole here of the gluteus maximus muscle, so the back muscle of the mouse. So this is the blood vessel, so we'll be looking at 2A arteriole. So the model, the experiment is when you apply vasodilator, in this case acetylcholine, it will cause vasodilation to the blood vessel. And you can measure this using the instrument shown here, the amplitude. So this is local response. And in conducted response, meaning that it's about 500 micrometer upstream, the amplitude is measurable, but it is slightly low. So the local response control would be just acetylcholine with saline, and then when they treated histamine, it doesn't do anything to the local response. So shown here, if you put chaga, mushroom extract, chaga, or chaga plus histamine, not a lot of changes in the local response. And in this case, you put E-tinctorium, no significant changes in the local response. So when Samreen measured the conducted response, meaning that about 500 micrometer upstream, histamine will prevent that vasodilation induced by acetylcholine. And this is, So this is consistent with what people know. Histamine, the way it does it is, so this is histamine. This is the proposed mechanism. I believe it has been shown in vitro, but I'm not sure about in an animal model. So histamine, the way it works, is that it binds to the receptor of the membrane, and then it causes signaling pathway, which ultimately leads to phosphorylation of the gap junction proteins, change in the structure, conformation, and then you lose that cell-cell communication. Okay, therefore, there is no vasodilation. But when she put chaga with histamine, you get vasodilation. So suggesting that chaga, the mushroom extract, can actually block histamine-induced effect. Same thing with E-tinctorium. So when she treated histamine-induced blood vessel with E-tinctorium, it can block the effect of the histamine. Okay, so these findings are important in two respects. One is it shows that whatever we found in vitro in the cell, it is actually happening in an animal setting, in vivo. And second is our in vitro model in screening for anti-inflammatory compound is valid. Right, so these are the current research that are ongoing in our lab. Almas is looking for trying to purify small molecule from Albatralis flatty. Vicky is looking at purifying and characterizing immunostimulatory compound from E. aurea, and Marin is looking at immunostimulatory compound from E-tinctorium and Ganosuge, and Brian is looking at immunostimulatory compound from Rauporus babeus. Okay, just 3 more slides to go here. So this slide and the next slide, I used it in my close-out meeting with Genome BC, basically telling them what we have achieved and so forth. Okay. So I thought I'm going to share this with you. It gives a perspective of what we have done, what we have achieved. So We have generated in total 540 crude BC mushroom extracts. Linda has put them into 96 well plates, and we have sent them to CDRD at UBC. So they're interested in screening for whatever bioactivities are ongoing at UBC. We have created a biological database containing genetic information and 3 bioactivities of 107 BC mushroom species. We have attracted new collaborations, academic and industrial. We got new infrastructure funding, and we also generating important information. We can educate the public and the government about the importance of preserving the ecosystem. And the strategic outcomes and benefit to BC and Canada. We're getting very good in doing in collection, extracting, screening for bioactivities, and we're also getting better in purifying and strategically identify bioactive compounds. And I believe we are an emerging team in natural product research discovery, and then we have generated biological database and library of those crude extracts. So they're now available to anybody in Canada who wish to use them. So where do we go from here? So this hypothesis that we set out to test, at the moment we are not able to answer this yet. So give us another few years after we have identified compounds, then we can see whether our hypothesis is true or not. So future directions, we need new money. Without money, words are just words. We won't be able to do anything. All right, so we need money to do the research. So I'm actively looking for them. So if we have the money, we'll continue on what we do. What we're doing right now, we'll use the grind and find approach in looking for these bioactive compounds that have potent activity and have not been studied. I would like to attempt to cultivate some of these edible and medicinal mushrooms that appear to be very exciting. Genome BC likes omics technology. So if we want to get money from Genome BC, we got to use omics technology. We got to collaborate with people in BC or across Canada to do that. So right now, there's an approach where people look at the proteomics and metabolomics data and be able to predict how a particular species make a particular compound. So something that we may venture into. And lastly, I'd like to collaborate with UNB researchers, for example, Sarah on the diabetes model, or Sean, Paul on their liver fibrosis model. So just a few pictures that we took. So Hook is holding on a precious Hydagoy Ganoderma suya. Okay, so this is the good stuff here. And yeah, we counted a lot of rains. Trip, yeah, it was the best part of the research, by the way. Trip. And yes, Linda and Maggie, they have to work overtime. So after we collected the samples, we gotta dry them. So it's work during the day and work at night. Okay, that's it. I can't take any questions. Yes.
Can I compare its like anti-inflammatory properties to typical anti-inflammatories like
No, we have not done the experiments yet. So, yeah, we have, I guess we ventured into anti-inflammatory because it's a very easy assay to do because we are looking at TNF production and then at the same time we look at inhibition. And NSAIDs, I guess, has a lot of side effects. So there's active research looking for new anti-inflammatory compounds. We have not compared them. Yeah.
Actually, that looks like a lot of work.
Yes, a lot of
Huge amount of work. It's amazing. And well, I come from Eastern Europe, so they do look into wild mushrooms and how to cultivate them. And sometimes it's tough because you need a certain type of tree, a certain type of dirt or whatever, with a certain type of rain schedule, things like that. So you said that the mushrooms in Hawaii were similar, like same species, but actually different than the ones on the continent.
In Prince Rupert, yeah, different.
Right.
Yeah.
What are the biggest challenges do you foresee in actually try to cultivate these? Because did you start looking into that or not yet?
Not yet.
No.
Not yet. Yeah. So I've been doing searches. Actually, people have cultivated Canada suge. There's a company in US that actually have these. You can buy it from them, and then you can actually. I think they're spores. So you buy from them, and then you can tend to culture them. Yeah. So people have done that for certain mushrooms, and others are not. So even chaga, people have actually cultivated them, and all the cultured chaga, they do not produce the chemicals that in the wild chaga. So people have actually scrapped the idea, and they actually just focus on collecting wild chaga. Yeah, you're absolutely right that some are easy to cultivate, but not others. And you're talking to an inexperienced person. I don't know anything about cultivating mushrooms. I would be collaborating with people to do that. Yes.
Yeah, that's really interesting. I know you talked about the work you did with Jeff to validate the findings. Both of those, I guess, are short-term effects. Do you have plans for sort of chronic, like treatment using those extracts?
Yeah. I've been looking into people with expertise in IBD diseases as animal model. Before we go into that, we want to be sure that it is a novel polysaccharide before we want to do a lot of work. So I think the bottom line is before we want to spend a lot of effort, money into expensive animal model, we want to be sure that the compound that we're working on is new. If it's something already known, there's less incentive to do that.
And then, sorry, just one more question. Yeah. So how often do you see, and you your table, but it's just how often do you see the effects without the growth condition? Like instead of the growth condition? Yeah, so it's really cell survival that you're measuring. So are you seeing a lot of those effects on growth condition? I guess I'm just thinking about if you were treating technically as something that's toxic and that's why it's killing itself, that's not necessarily a medicinal compound. So you see that, do you see that often when you don't have it?
Yes, to me, MTT assay is actually an accepted model for looking at growth. Yeah, from what we have seen, it's looking at mitochondrial staining. So live cells has healthy mitochondria. Dead cells, they don't have healthy mitochondria. So although we have seen correlation between growth and inhibition of growth and staining of MTT. So yeah, in terms of toxicity, I know. So we haven't gone into animal models. So in terms of toxicity to animals, we don't know anything about that yet. But in terms of cells in petri dishes, I don't think one could distinguish between toxicity and growth inhibition. Alu?
I'm curious about the fact that look at pro-immune properties and anti-inflammatory properties, which are kind of protein. And you look at TNF-alpha as a molecule. So did the in vivo histamine work, which helps to kind of, I guess, establish the inflammatory aspect of that a little bit more? But what other targets would you look at down the road? Trying to part a little bit more immunomodulatory and
Yeah, okay, yeah, that's a good question. TNF alpha, we just look at TNF alpha as a screening because it is widely used and accepted in the field. Okay, yeah, and we actually found that it is very sensitive. TNF alpha production is very sensitive. We have actually looked at production of other cytokines since, like interleukin, Th1, Th2 for the responses. They are in use, right, in use as well. Yeah. So down the road, if we actually pick a compound that's novel, and when we look in an animal model, we would like to look at animal model, and we would actually look at not TNF alpha, right, but a bunch of cytokines. Yeah. It's more like a first screening method to look at whether it actually can stimulate immune cells. TNF alpha. And in terms of anti-inflammation, what we've done so far is more like quick, easy to see if there's an activity that's relevant to that aspect. Thanks.
Yeah, Chow, sort of in the same theme, did you find any correlation between any of your three variables? Like, did the growth inhibition, some of those fungi also have stimulatory effects, or not stimulatory, but anti-inflammatory?
No, there's no correlation. So in fact, actually, those that stimulate the immune system, strongly stimulate the immune system, they have weak inhibitory growth inhibition. We found that quite the opposite.
What about the anti-inflammatory?
Now, when an extract has no immunostimulatory activity, then we use those to do the screening for anti-inflammation. Those that has immunostimulatory activity, we don't typically test them for anti-inflammation.
One other follow-up question. One of our big problems when we were in the field was obviously collecting enough material of the same fungus to do the research. And that was a pretty big block for some of the things we were finding out there. So do you see any progress in that area where you might need less substrate to be able to do some of the extracts?
For sure, yeah. So I know there's a mushroom that are about this big, and we got to collect hundreds of them, so it wouldn't be on top of our list unless it is unique. Nobody has worked on it, and then maybe we'll try to cultivate. So it's definitely not something that we want to jump into it, but we have to think carefully, because the fact that we have so many to choose from, so we have to prioritize this.
So we still need quite a bit of substrate in order to proceed with extraction.
It depends on, for bioactivity guided research and purification, like what we do, yes. I was in a lab in Griffith University. It's a natural product research lab. They are chemists. So chemists would do it differently. So they would actually just look at, they have a high power LCMS, NMR at their fingertips. So they would be looking at novel compounds. Yeah, and that wouldn't require a lot of material. So I'm still learning that, learning from a chemist, how they approach this area of research.
Is there any other questions? Well, Chow, thank you so much for giving your talk today. I think this is exactly, just sort of shows how research technologists can really spark a lot of systems that work progressive and do end up progress. So thank you so much.
Thank you.
Thanks.
Let's not look forward to hearing about your